There hopefully it's audio. I just haven't started talking yet as I'm waiting for people to get on. Do you hear me, Andy? I guess I should probably play music in the background in the beginning. I don't know what's the best song to play, but Beethoven, that would be a good idea. Dvorak, Dvorak, Bach. So classical music, okay. Feeling alive, that sounds good too. Um, Good morning, all of you. I hope you are all doing well and your families are staying safe. um, Today, we're going to move on from chapter 16, where we covered gluconeogenesis, to chapter 17, where we look after the citric acid cycle. Let me just go and grab a quick drink here of water. Um, in the chapter 16, we went and took a look at how glucose went to pyruvate and also how pyruvate went back to glucose. And again, these were things that took place during anaerobic conditions. What we're going to look at now is what takes place during aerobic metabolism. Aerobic metabolism meaning in the presence of oxygen. And so during aerobic metabolism, pyruvate is converted to CO2 and water. And in this process, they generate, in effect, reducing equivalents of NADH or FADH2. This NADH and FADH2 then goes on to produce ATP. Also during the process, you also produce some NTP slash ATP directly. And the first part of this is a citric acid cycle. And really, there's two parts to the citric acid cycle. In the first part, there's two parts to chapter 17, I should say. In the first part, you convert pyruvate over to acetyl-CoA, and then you go from acetyl-CoA to convert that to CO2 and reducing equivalents. And that takes place in the actual citric acid cycle itself. Today, what we're going to mainly focus on is the conversion of pyruvate to acetyl-CoA. In chapter 18, we learn about how the NADH and FADH2 produced in the citric acid cycle is used to go and generate ATP. And I should point out that this takes place inside the mitochondria. Glycolysis takes place inside the cytoplasm. So this takes place in a separate location from where glycolysis takes place. So without further ado, let's go and take a look at the citric acid cycle. And so the citric acid cycle is sometimes called the TCA cycle or the tricarboxylic acid cycle because there's tricarboxylic acids in the cycle, or the Krebs cycle because Krebs was the person that elucidated it. And again, this requires oxygen. So this is what they call aerobic metabolism. Initially, you go from pyruvate to acetyl-CoA, and then the acetyl-CoA goes around the cycle itself, which takes place inside the mitochondria. And ultimately, the NADH that you produce is going to produce a proton gradient, which is then used to produce ATP. But that's in chapter 18. So shown here is the first part where we go from pyruvate to acetyl-CoA. In this case, you lose CO2 and you generate two high energy or two electrons or two reducing equivalents. And so this is what you're going to go and produce. Now this is done by a pyruvate dehydrogenase complex. And as the name implies, it is a complex. It is quite complex. So we're going to go through how this complex works and go from there. So one of the important aspects of this is the product, which is acetyl-CoA. Shown here is the structure of acetyl-CoA. The only part that we're really going to focus on here is going to be this part over here. So please make sure you're aware of the thioacetyl group and aware that that's connected in such a manner. You also should have some, I mean, the rest of the structure is pretty simple for you at this point, but please just remember acetyl-CoA. Shown here is the mitochondria. The mitochondria looks a lot like a bacterium. And in many ways, people have proposed that this, in fact, is a result of a eukaryotic cells engulfing a bacterium. And so it has an outer membrane and it has an inner membrane. And you have the outer mitochondrial membrane and the inner mitochondrial membrane and the matrix. And just like in a bacteria, uh, most things go through the outer membrane and very few things go through the inner membrane into the matrix. The interesting thing about mitochondria is that it does contain DNA, and it's really pretty much the only place besides the nucleus that you're going to find much DNA. And it actually does go and have its own genetic code. It's a little bit different from that that's in the nucleus. And it does produce a few proteins. It doesn't produce very many, but it does produce a few. And this is, yes, it is the powerhouse of the cell. That is correct. And that's where you're going to produce a lot of ATP. And we'll go into that in chapter 18. Shown here is what happens to the acetyl-CoA. The acetyl-CoA is listed here as C2 because the acetyl group has 2 carbons in it. It then combines with oxaloacetate, which is a C4, to make C6. And as you go around the cycle here, what you see is you produce a lot of reducing equivalents and CO2, and you also produce 1 GTP, which can then be used to go and provide energy elsewhere. What's important here to realize is that these NADHs and FADH2 ultimately will go on and produce ATP through oxidative phosphorylation, which we'll cover in chapter 18. You can see that in the next slide here. Shown here is the citric acid cycle, how you produce 8 electrons. Each NADH carries 2 electrons, and each FADH2 carries 2 electrons. So 3 NADHs, 1 FADH2, that makes 8 high energy electrons. These then go through the electron transport chain, which we cover in chapter 18, and produce a proton gradient. And this proton gradient then is used to produce energy in the form of ATP. So the first part we're going to talk about is the formation of acetyl-CoA from pyruvate. And this takes place under aerobic conditions. Although you don't see oxygen here, the reason it's under aerobic conditions is ultimately you need some place for the NADH to go. And so it is regulated in such a manner that it only will take place in the presence of oxygen. And initially, you have to get the pyruvate into the mitochondria, and it's done through a transporter in which it's exchanged for OH minus. So pyruvate goes in, OH minus goes out. This is called an antiporter because things move in opposite directions. And this is the driving force to drive the pyruvate into the mitochondria. This reaction is irreversible in us. And so that means that once we go from pyruvate to acetyl-CoA, there's no going back to sugars. We cannot go back and produce sugars from acetyl-CoA. So pretty much once you've gone from pyruvate to acetyl-CoA, you're not going to produce, you'll be able to use gluconeogenesis to go back and produce glucose. So at this point, you can use the acetyl-CoA to produce fat, but you can't use it to go and produce sugars. And this is really the link between glycolysis and the citric acid cycle. Shown here are the components of this complex. It is a big complex. It has an molecular weight of 4 to 10 million Daltons. Daltons is just a fancy name for grams per mole. There's a subtle difference in definition, but ultimately is pretty much the same thing. And so therefore, you have 4 to 10 million Daltons. So this is something of a molecular weight of 4 million to 10 million. So it is big. And it contains three main components that we're going to discuss. One is the pyruvate dehydrogenase component, which is separate from the pyruvate, part of the pyruvate dehydrogenase complex. It also contains the dihydrolipoyl transacetylase and the dihydrolipoyl dehydrogenase. But most people do this really simply. They call them E1, E2, and can anybody make a guess what the next one's going to be called? Oh, I was seeing if people were E. That's right, E3. And so therefore, what happens here is that you go E1, E2, and E3, and this complex contains multiple E1s, E2s, and E3s. And E1 catalyzes the oxidative decarboxylation of the pyruvate. Then you get transfer of the acyl group to CoA, and then regeneration of lipoamide. And we'll show you these two other important cofactors. So other important cofactors in the pyruvate dehydrogenase complex are TPP. TPP, electronic entertainment expo. Yes, that works well. As shown here is TPP. The most important part of the TPP is the central core part here. That's the thiamine. And so therefore, it's very important. And then you also have the five-membered disulfide ring here. So both the disulfide ring and the thiamine ring are important in this particular process. And so you should know what those structures are and understand what's going on there. So shown here is kind of in schematic form what takes place. We'll go into it in much more detail in the next few minutes. But what happens here is you start with pyruvate, you release CO2, and you get, in effect, a carbonyl group here and a negative charge here. You transfer those electrons over to ultimately to NAD to make NADH, and therefore you then get now a positively charged acyl group, and then that can be transferred over to CoA. The first two steps are done on E1. The last step is done on E2, and we'll go into exactly how those steps take place. Where is the minus charge? The minus charge is in theory on this carbon here. And so really, it's actually, it should be really, truly, there's not really a carbon there. We'll get into that in a second, and we'll show you where that is. But it's kind of just, in theory, it's really just kind of hanging there, to be honest. Shown here is TPP. TPP, again, is the cofactor. And the nice thing and very strange thing about TPP is that this proton here has a very has a very uh, low, relatively low pKa. Most CH groups on carbon have a pKa that's going to be, somewhere around, in the case of a hydrocarbon, somewhere around 50. But this here has a pKa that's around 10 or so. And so, given a pKa of around 10, what that means is that it's going to be quite acidic, and you'll be able to make the corresponding anion. This anion is actually on that carbon there, and then you make H+, and so therefore you can make TPP anion relatively easily. The TPP anion, like many anions, is going to be quite nucleophilic. And so what happens in this reaction mechanism is the anion of TPP that's on that particular carbon here that's on this carbon here is going to go and attack the carbonyl group of pyruvate. It makes what they call the addition compound. The addition compound then goes and loses CO2. And this gets you the hydroxyethyl TPP. And hydroxyethyl TPP is shown there. And so shown here is hydroxyethyl TPP. And so really, you lose a CO2, you generate this hydroxyethyl TPP with this anion here that's going to be important. There's a negative charge right there, so let me just put that negative charge and circle it. If you pronate it, you get the hydroxyethyl TPP. But what's important is the resonance form of the hydroxyethyl TPP. This negative charge here, of course, is going to be quite nucleophilic, and we'll see how it goes and reacts on the next slide. So let's go and take a look at the next slide. So this negative charge on the hydroxyethyl TPP reacts with the disulfide of lipoamide. It then goes and loses the TPP, and you make the corresponding acetyl lipoamide. And this step here is very important to understand that you're actually oxidizing a hydroxyethyl group and you're reducing the lipoamide. And so what happens here, of course, is this here on the left side is equivalent to a disulfide. Well, over here on the right side, we have two, in effect, thiol groups. So what we've done is we've taken a disulfide bond and reduced it down to a thiol. And so by taking a disulfide, reducing it down to a thiol, that's going to be a reduction. On the other hand, what we've taken here is a hydroxy group and we made a carbonyl group. So in going from the hydroxy group to the carbonyl group, what we've done is we've done an oxidation. So therefore, what happens here is the hydroxyethyl group gets oxidized to an acetyl group, while the lipoamide gets reduced down to the reduced form, or to acetyl lipoamide, which in effect is the reduced form of the lipoamide. And so it's both an oxidation reaction and a reduction reaction, as is the case for all redox reactions. And again, that's what's taking place here. Once you've generated the acetyl lipoamide, what it does is it transfers the acetyl group to CoA to make acetyl-CoA. This is the third step. And in this case here, this reaction is catalyzed by the E2, which is called dihydrolipoyl transacetylase. And now you see why it's called a transacetylase, because it transfers an acetyl group from lipoamide over to the coenzyme A. And that's catalyzed by E2. Now the problem is, in order for this to go and actually happen again, the dihydrolipoamide has to go back again from the reduced form, which is dihydrolipoamide, to the oxidized form, which is lipoamide. In this case here, you transfer the electrons to FAD to make FADH2. The FADH2 then transfers to electrons NADH or NAD to make NADH. So ultimately, you're transferring the electrons from dihydrolipoamide over to NAD to make NADH. The thing that's very strange here is that normally FADH2 is not strong enough to go and reduce NAD down to NADH. And that normally does not take place. But because the FADH2 is bound to the enzyme and as a prosthetic group and is not going to leave, what happens here is that it is in such a conformation that it actually has a higher reduction potential, so it can reduce NAD down to NADH. And that's again because of the conformation of the FAD within the enzyme. Shown here is the lipoamide. And what you notice here is that the lipoamide is attached to the lysine residue here on E2, and it goes and makes a very long attachment point. And so the idea behind this that's very important to understand is that this is the same concept as biotin. In biotin, you had a long arm, and the biotin could go and move around when you were going and doing the pyruvate carboxylase. In this case here, you have the lipoamide on the end of a long arm, and again, this arm can move around. And so in effect, what it does is it moves around the pyruvate dehydrogenase complex from E1 to E2 to E3, but it's actually always covalently bound to E2. So this is the same sort of thing as a production line, where you take one component from the first step to the second step to the third step. And so that's what goes on there. You can see here a picture of the pyruvate dehydrogenase complex containing the three components. And please note it contains multiple numbers of each of these three components. So it contains many E1s, many E2s, many E3s. E2 is in the center, and that's not surprising. If it's going to act to go and move from one to the other, it then has access to E1 and E3, which are on the outside. Shown here is my favorite picture. In this case here, what you have is you have the TPP, which is going to be bound to E1. E1 is shown in yellow as a yellow ball. You have FADH2, which is part of E3, which is shown here in green. And then here you have your E2, which contains the lipoamide side chain, and that's shown here in, I guess, pink or magenta, depending upon what you like. I don't know. Everybody has depending on what it looks like on your computer. So step number one here is going to be dictated by the TPP. So this is where TPP goes and reacts with pyruvate to lose CO2 and create the hydroxyethyl TPP. You can see hydroxyethyl TPP there. The lipoamide then goes into the active site of E1. The lipoamide is covalently attached to E2, but it goes to the active site of E1. Once it's in the active site of E1, it can go then and have the hydroxyethyl group transferred over. And so therefore, you then go and get the acyl lipoamide. Once you have the acyl lipoamide, that then can transfer the acyl group or acetyl group over to the CoA. That gives you the acetyl CoA, which is shown here. And so this group here is transferred over to CoA. Once you've done that, you need to go and, in effect, oxidize this reduced form of or the reduced form of lipoamide, which is dihydrolipoamide, back up to the disulfide. That takes place in E3. And then you could get back to where you started all over again. And so this is a very nice schematic, which just shows you the various steps. It also shows you the various cofactors that are involved here. You have, of course, TPP, FAD. You're going to have, of course, CoA. You're going to, of course, have FAD as well. And you're going to have NADH or NAD+. And so you can see here that there are many cofactors that are involved in this process of pyruvate dehydrogenase. Shown on the next slide is kind of the idea that you go from pyruvate to acetyl-CoA. And then once you get your acetyl-CoA through the pyruvate dehydrogenase complex, you then move on to the citric acid cycle. Shown here is the citric acid cycle, which we're all have some familiarity with. Please, again, just as you did for glycolysis, please go through the citric acid cycle and learn the various enzymes involved and know the various structures that are there. And so that way, and also ultimately how it's regulated and the various mechanisms, just as we asked for glycolysis. Now, what we're going to go and do now is go back and take a look at the worksheet for the pyruvate dehydrogenase complex. So what I'm going to do now is give everybody off to a breakout room for, let's say, 20 minutes, so to 11:45. And what we'll do then is we'll go and we'll do the pyruvate dehydrogenase worksheet. So let me go and set up the breakout rooms now. If you don't get assigned to a breakout room, please please let me know, and I will go and take care of that. So right now we have, oh, thank Brittany, who's even faster.
How are you guys doing? Hello. Good, good. Hello. All right. So I'm gonna give you guys like 5-10 minutes to do the worksheet on your own, and then we can go over it together. Does that sound good? All right. And let me pull up the worksheet. And if you have any questions, meanwhile, you can always just let me know. Sure. Is everyone done or almost done? All right. How about 2 more minutes? Does that sound good? Okay, sure. All right then. All right. Give me 1 second. Okay, so how do you guys find this worksheet? Easy, medium, hard. Yeah, it is pretty straightforward. It's just that you have to know basically the information about pyruvate dehydrogenase complex to proceed, because that's what this entire worksheet is about. It's very focused on that dehydrogenase complex. All right, let's get started then. So how many cofactors do we have? Mm-hmm. What are they? Okay. Mm-hmm. And if you guys don't know where to get those, you can always go to the PowerPoint. NADH, lipoic acid, and acetyl-CoA. Exactly. Okay. Now, drawing them out is going to help you answer B and C. So again, if you need help drawing the cofactors, you can always go to the PowerPoint. They're all drawn over there. So let me give you one example, like TPP. Were you guys able to find the structures or no? Okay. Would you like me to go over them? In a PowerPoint, it will be slide number 4, 6, slide number 10. This is where you would find, yeah. Or you can find them in the book as well. Like TPP, for example. Because they did ask to draw the ring structure. So you don't have to draw everything, you just draw the important ring structure. But of course, knowing the entire structure could be helpful because you don't know what you'll be asked on it. Oh, Nepil. Yes. Acetyl-CoA is the one without a ring. So that's TPP, basically. Then lipoic acid. Oops. You would just have a sulfur bond here. And that's like a carboxyl group at the end. And notice that this is a disulfide. and it can be reduced to a dithiol. And just very important to note, this here is very acidic. Acetyl-CoA, it does not have a ring because that's how it looks like. That hydrogen, let me draw it up. This hydrogen here, that hydrogen is very acidic. Yeah, no, I was talking about the TPP. Here, there's a disulfide in glycolic acid. Okay, and then NADH, FADH2, that should be a given, but do you guys want me to draw NADH and FADH2 as well? Okay. I think, yeah, I'm gonna have to erase. So, yeah, just honestly look it up, basically. It's just that if we have time, I'll get back to drawing NADH and FADH2. It's just that I kind of want to touch on these three because I want to make sure you guys can know where to find the answers to these. And then I'll go back to NADH and FADH2. Does that sound good? All right. Okay, so what three main subunits make up the pyruvate dehydrogenase complex? B and C, yes, I forgot about those. Okay, so how many, based on the structures that we've seen, how many of these cofactors are covalently attached, do you think? What do you think they are? All right, lipoic acid for sure. FADH2. What else? And TPP. Let me check something. Now here's the thing about TPP here. If you look at the mechanism, so if you go to slide 2, 3, 4, 19. Slide 19. You guys want me to pull up the slide? Yeah, it will be FAD. Yes, sorry about that. Yeah. If you look at the slide, you'll notice that NADH dissociates, right? Acetyl-CoA. So that mechanism is the answer to question number G. A schematic direction indicating the movement of molecules between the active sites of the three main subunits. That's the answer to G. You do have to know that entire slide. And then the mechanism using errors of the pyruvate dehydrogenase complex. That's the three-step mechanism, which is it starts in slide number 11. So this is slide 11. It's like it starts with the reaction consists of three steps: decarboxylation, oxidation, and transfer of the resultant acetyl group to CoA. Not slide 3. I'm referring specifically to slide 9. It's a three-step mechanism. There's decarboxylation, oxidation, and transfer to acetyl-CoA. That's where it starts. And then the mechanism in detail is in the 3 slides afterwards. That's for H. For G, it's that schematic. It's the circles and all these things moving around. That's the one for G. 6, 8, 9, 10, 11, 12, 13, 14. Show you the slides? Okay. Thing is, I'm gonna have, let me quickly take pictures. Oh. Well, just remember that it's in the PowerPoint.
Good morning again, all of you. I hope again you're doing well. I was just going to go over the worksheet very quickly with you and then go on and continue on with the actual cyclic portion of the citric acid cycle. So how many cofactors does pyruvate dehydrogenase complex use? It uses 5, and that's going to be considered TPP, FAD, NAD, CoA, and lipoamide. And these are all cofactors. How many of these cofactors are covalently attached to the complex? We're all familiar with lipoamide being covalently attached to the lysine. The flavin is also covalently attached. And so therefore, there are actually two, they're going to be covalently attached, and then three that can dissociate. So we have NAD, we have, which clearly goes on and off. We have clearly CoA. For acetyl-CoA, it goes on and off. And then it turns out that TPP is not covalently attached, and so therefore there would be three. In the case of D, list the cofactors below. We've listed the cofactors up above, and so again, you would just need to list, TPP, FAD, NAD, CoA, and POMID. Again, here, draw the important ring structure. So clearly, TPP has an important five-membered ring structure, and so you would need to draw TPP. FAD has an important ring structure as well for the oxidation reduction, where we go for there. And so therefore, in this case here, covalent would be going to be FAD and going to be the lipoamide. Here we're going to get NAD. We're going to also have CoA, and TPP is not so clear. And so, although it's not covalently bound, let's just put it in the middle because that was not so clear whether it was covalently or not covalently bound. The important ring structures would be TPP, the also FAD, the NAD, the six-membered ring for NAD, membered. You have the three rings for FAD, the five-membered ring here. And then you would also have CoA clearly is not going to have a ring structure, and so that one was excluded. And lipoamide has a five-membered ring. And so you need to draw those particular structures. So, so covalently attached are going to be FAD and lipoamide, and then the ones that are not covalently attached are going to be NAD and CoA. The one that's a little unclear is TPP. I did write it on the left side. I don't know if that's not showing up or not, but let me know if there's problems. This here is a really difficult one. What three subunits make up the pyruvate dehydrogenase complex? This one's actually not difficult at all. It's just E1, E2, and E3. Those would be the common names for them. You should know that it's, pyruvate dehydrogenase component, the, transacylase, and also the lipoamide dehydrogenase. And so therefore, dihydrolipoyl dehydrogenase. So you should know what the names are. The schematic would be just the same one that we showed you on the slides, where we showed you the arrows moving to go and going from TPP over. And oh, sorry, I apologize. I was thinking of the next one. G here, this is just the one with the circles where we have, E1, E2, E3, E2 and E3. And you clearly show that, each one with the corresponding cofactor. And so again, this is from the PowerPoint, just to make sure everybody understood what the important components were. And then the mechanism here would be one, again, from the PowerPoint with the actual arrows drawn. And so that would cover the worksheet. I'll go back to the top here for 1 second because I expect somebody will ask. But what we're going to do now, having done the first part here of how we go from pyruvate to acetyl-CoA, what we're going to do now is we're going to go and take a look at how we go from acetyl-CoA through the citric acid cycle. So as you can see here, we have our citric acid cycle. Citric acid cycle is not as bad as glycolysis. There are only 8 steps as opposed to 10. In terms of naming, most of these steps, enzymes are relatively easy if you know the structures. I mean, we have malate dehydrogenase, we have isocitrate dehydrogenase, we have succinate dehydrogenase, and we have alpha ketoglutarate dehydrogenase. And so all of these 4 enzymes are just named in the standard traditional dehydrogenase manner, in which you name them after the most reduced species. So these names are very easy. You have 2 synthases or synthetases, and so again, that's relatively easy. It just tells you what they make. And then there are 2 ones that are, I would say, a little harder, fumarase and aconitase. And so those are the 2 ones that are going to be a little harder. But again, in terms of the big overview, what I would like people to note is, again, there's 4 dehydrogenases. And so this is all about getting electrons out of the acetyl-CoA. The other thing I would like people to know is that it is a cycle. And so you just keep on going around and around. You put in acetyl-CoA, and the acetyl-CoA you put in shows up as the green atoms. So you want to be able to follow the green atoms around the circle. The green atoms will in fact represent what was put into the system. The green atoms stop at succinate, and the reason they stop at succinate is because succinate is symmetrical, and so therefore, in theory, you cannot distinguish what, if you labeled the green atoms, the label would just be, in effect, dispersed. So if you labeled the, for example, if you labeled, let me just, if you, for example, labeled this carbon here, that's going to end up here, 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 and then it's going to end up here, and half here. Because again, you can't distinguish between the 2 CO2s, and therefore, which mechanism? We'll tell you in a minute. So please hang on for that. And then if you did the circle here with the CH3, then that would end up here, this one here, this one here, this one here, this one here, and then it would be equally dispersed between these 2 CH2s because again, it's symmetrical. There's no chirality there. It's not prochiral, and so you can't see it. So interestingly enough, when you go through, when the acetyl-CoA goes through the first time, you don't actually lose those atoms as CO2. You lose other atoms as CO2. So the atoms actually, you lose a CO2 come from oxaloacetate as opposed to acetyl-CoA. However, if you keep on going around and around the cycle, then clearly the only input of carbon is acetyl-CoA, so ultimately they will lose as CO2. So that gives you an idea of the particular mechanism. The other thing to note here is kind of why do these steps occur? So, the first thing to see here is that you want to, in fact, get out reducing equivalents, and you do that in 4 cases here. And you also want to go and lose CO2. So the first thing you do is combine oxaloacetate and acetyl-CoA to make citrate. You have an intermediate of citryl-CoA, which loses the CoA to make citrate. Now, the problem with citrate is that citrate, the alcohol in citrate is what? A primary alcohol, a secondary alcohol, or a tertiary alcohol?
Tertiary.
Tertiary, that's right. It's a tertiary alcohol. And as we all know, tertiary alcohols are very hard to oxidize. And since they're hard to oxidize, what happens is that you have an enzyme called aconitase, which in effect does a dehydration reaction and then a rehydration reaction. So you do elimination reaction, then you do an addition reaction. In the case of aconitase, what happens is the OH goes from being a tertiary OH over here. Oh, what did I do? a tertiary OH over here to being, what is the OH in isocitrate now? Is it tertiary? Secondary, that's right. It's secondary. And because it's secondary alcohol, you can now oxidize it easily. Lo and behold, the next step in the pathway, you take that secondary alcohol and you oxidize it up to a corresponding ketone. Now, in the process of oxidizing it up to a ketone, you spontaneously lose CO2. And we're all familiar with this from organic chemistry, where we went and studied uh, I guess, ACAC synthesis, malonyl ester synthesis. So we take malonate, we lose CO2. We also studied acetoacetate syntheses, where again, you would automatically lose CO2. And so the idea here is that when you have a molecule that looks like this, I was going to draw in the acidic form. What happens here is you lose the CO2 that's beta to the carbonyl. So shown here is the carbonyl. This is alpha, so you don't lose that CO2. This here is alpha, this is beta. So this here is the CO2 that you're actually going to lose. This is beta, this is gamma, and you don't lose the gamma CO2. And so therefore what happens is you spontaneously lose the CO2 in the beta position, and you end up here with alpha ketoglutarate. And the thing about alpha ketoglutarate here is that alpha ketoglutarate looks a lot like something else we've seen before. Let me just erase all of this now. This alpha ketoglutarate looks a lot like pyruvate. The only difference is in alpha ketoglutarate, you have this group here. In pyruvate, you have, I guess if I'm going to make them parallel, you have this here. And so therefore, that's the only difference. And so just like you have a pyruvate dehydrogenase complex that goes and converts, in effect, pyruvate to acetyl-CoA, you have an alpha ketoglutarate dehydrogenase complex that does exactly the same thing. So the alpha ketoglutarate dehydrogenase complex has exactly the same strategy as the pyruvate dehydrogenase complex, which you already have studied already. The alpha ketoglutarate then loses, again, CO2, and you generate succinyl-CoA. Again, this is equivalent to what you did with acetyl-CoA. So this is very similar to acetyl-CoA, S-CoA. Except for instead of the CH3, you have these groups here. So you generate succinyl-CoA. Now, as we mentioned before, thioesters are energetically very similar to the amount of energy in ATP. And so what happens here is that this thioester is then used as a hydrolyzed thioester. You convert, in effect, generate GTP, which is another NTP as opposed to ATP, but you generate the GTP. So you use the energy in the thioester to go and generate succinate. Once you get to succinate, again, you want to generate these high energy electrons, so you make FADH2. It turns out that there's not enough energy, that succinate is not reducing enough to be able to reduce NAD down to NADH, but it is able to reduce FAD down to FADH2. And that's why you don't use NAD, that's why you use FAD. Once you make fumarate, you then just add water across the double bond as fumarase. You make malate, you oxidize malate up, again, to get high energy electrons out, and then you end up with oxaloacetate, and the cycle goes on. And so that's how the cycle works. That's a generic overview of this particular pathway. So again, here, shown here is the first step in the citrate synthase. You just combine acetyl-CoA and oxaloacetate. You make citryl-CoA as intermediate. In order to drive this reaction forward, you hydrolyze the thioester to make citrate. Because as I mentioned before, bringing two molecules together is generally very difficult. And so that takes energy. And the driving force for this reaction is the hydrolysis of the CoA. And so therefore, this makes sure the reaction transpires and occurs. Shown here is the structure for citrate synthase. And again, just to make sure you don't actually hydrolyze the acetyl-CoA, it's the same way as we saw with kinases. What happens is it goes and has order kinetics. It binds the oxaloacetate first, and only after the oxaloacetate binds will actually form the binding site for acetyl-CoA. And this is to minimize the amount of acetyl-CoA that gets hydrolyzed, just as most kinases bind the substrate first and then bind ATP afterwards, because that way you form the binding site. Shown here is the mechanism for this reaction. It's not a complete mechanism, but it is a schematic mechanism. Please, again, make sure you know these particular mechanisms. In this case here, it's a pretty simple mechanism. The first thing to do is you go and activate this carbonyl through a NH here, and you remove a hydrogen here to go and make the enol intermediate. This enol intermediate then goes and attacks the carbonyl of oxaloacetate in a standard condensation reaction, and you make the corresponding citryl-CoA. And this is very similar to reactions you've seen in organic chemistry. And so there's nothing really special here. It's just catalyzed by an enzyme. The next step is to go and isomerize the citrate to isocitrate. Again, this occurs because you can't really oxidize a tertiary alcohol, so you make it into a secondary alcohol. The reason the enzyme is called a conatase is that the intermediate in this is, in effect, is cis-iconatate, which is the name for that particular compound, and so the enzyme was called a conatase. Shown here is the structure of a conatase. It uses an iron-sulfur cluster. The iron-sulfur cluster goes and binds to the citrate. It then catalyzes the elimination of the citrate to make the iconatate, and then you add water back in to make isocitrate, which is a corresponding isomer. And again, this uses a 4-iron, 4-sulfur cluster. The reason it's called 4-iron, 4-sulfur is it contains 4 irons and 4 sulfurs. You also get, 2-iron, 2-sulfur clusters, and so on. And so based upon the name, you know how many various atoms it contains. Shown here is the next step. And again, in this case here, you oxidize the isocitrate to oxalosuccinate. This oxalosuccinate then spontaneously loses the CO2 here, which we discussed because it was in the beta position, and you get beta decarboxylations relatively easily. The reason for that, of course, is if you want to draw a mechanism. You can go easily see how you can get this happening here. And so, well, I did something wrong here. 1, 2, 3, 4. Oh, that's because I 1, 2. I put in an extra. That was my fault. Let me do that one more time. And so this happens here. You get this sort of 6 structure here. And so therefore you lose the CO2 and you generate the corresponding enol. Enol then goes back to the corresponding keto through enol-keto tautomerization. And so in that case there, you generate the alpha-ketoglutarate. Again, this happens even in solution spontaneously, but it's actually catalyzed by the enzyme. Once you've done that, you go from the alpha-ketoglutarate to succinyl-CoA. This is just the pyruvate dehydroxylase complex, the hydrogenase complex all over again. The mechanism's the same, the strategy is the same, the cofactors are the same, and so therefore, and the result is the same. So that's what goes on here. Once you make the succinyl-CoA, as we mentioned before, you have a high-energy thioester intermediate. This then thioester intermediate then can be used to generate GTP. In some organisms, it actually makes ATP. And so therefore, and in some organisms, the enzyme actually uses ATP or GTP. And so therefore you make an NTP equivalent. And so again, you use the high energy in this thiol-ester linkage here, and you go and make the corresponding NTP here. And so that's what takes place. And so this way you get actually some energy out directly from the citric acid cycle. Most of it actually comes otherwise, but you can get it out directly that way. Shown here is the mechanism. Again, please go take a look at the actual mechanism. This one is relatively simple. You go and in effect take a histidine, you phosphorylate the histidine, and then you transfer that phosphate over to the NDP to make the NTP. And so it's relatively simple sort of mechanism. Shown here are the last few steps. In the last few steps, what happens here is first of all, you go and make the double bond. That's through succinate dehydrogenase. And so again, you use NAD. You don't use NAD, you use FAD because it's just not energetically reducing enough to generate the NADH. And so you generate fumarate. The fumarate then goes and gets water added through fumarase, and then you make malate dehydrogenase to go and do the corresponding oxidation. The stoichiometry of the citric acid cycle is shown here. Again, the thing about this is that you see no oxygen. But what's very important to realize is that in order for these NADHs and FADH2 to be regenerated back to NAD and FAD so the cycle can continue, you need oxygen. So although the mechanism doesn't contain oxygen directly, in order for it to continue, you do need oxygen around. And we'll show you how this NADH and FADH2 goes on to react with oxygen to produce water in chapter number 18. So again, please make sure please make sure that you take a look at this and make sure that you understand the enzymes involved and that you understand the structures involved in the citric acid cycle. Shown here is the energetics of the citric acid cycle. Again, please note, this is not delta G, this is delta G0. And so delta G0 does not take into account the concentrations found in vivo. Because if this was in fact delta G, you would be in big, big trouble. And you'd be in big, big trouble because the last reaction, number 8 here, has a very high positive delta G0 value. And so again, that does not take account what goes on in vivo. And so therefore, in vivo, this reaction actually does take place, in part because the concentrations are such that it is able to do so. And so therefore, please note again, delta G0 prime, not delta G prime. So therefore, that's important to understand. How are all of these in equilibrium? Well, in effect, most of them are going to be in terms of delta G0, most of them, only a few of them are in equilibrium. So these ones here, 0.09, 0, 0.08, maybe even 2. So these 5 would be in equilibrium. The other, maybe even this one here, sorry. The other 3, the citrate synthase, alpha ketoglutarate dehydrogenase complex, and malate dehydrogenase, again, would not be in equilibrium based upon the delta G0 prime values. But please note, traditionally people just draw them with equilibrium signs when they draw reactions. I don't know why they didn't do it for the first one, but in general, they should also do it actually for they're going to do it for the first one, they should also do it for the third one, which they don't do. But I think that's just a typo, and I wouldn't worry about that too much. But please note the 3 that are going to be problematic. The first, number 1 and number 4 are going to be very negative, while number 8 is going to be positive. But again, this is delta G0, is not delta G. So it doesn't take into account what's found in vivo. The site of regulation for these pathway or for the citric acid cycle is going to be the pyruvate dehydrogenase complex, diisocitrate dehydrogenase complex, and the alpha-ketoglutarate dehydrogenase complex. So those are going to be the three places that is going to be regulated. We can imagine that the alpha-ketoglutarate dehydrogenase complex is going to be regulated similar to the pyruvate dehydrogenase complex. And so we'll discuss that first and then go on from there. So the synthesis of acetyl-CoA is regulated by the pyruvate dehydrogenase complex. This is very important because once you go and make acetyl-CoA, you cannot go back to making glucose. And so therefore, this is relatively irreversible step and therefore is going to be relatively highly regulated. And so the complex itself is regulated allosterically and by reversible phosphorylation of E1. It's also regulated by, and this occurs by a kinase associated with E2. The kinase is regulated by NADH, acetyl-CoA, ATP. The phosphatase is stimulated by calcium. Okay, fine. Why might calcium be important? Why might calcium be important? Where is calcium used in the body besides bone? Muscles, that's right. They're used in the muscles. And it's used as a signal for muscle contraction. So therefore, if you want your muscles to contract, they're going to need lots of energy. And so for the muscle to contract, they need lots of energy. So you want a active pyruvate dehydrogenase complex. So the unphosphorylated species of the pyruvate dehydrogenase complex is active. And so therefore you want the phosphatase to be very active. So that way when your muscles get calcium and told to contract, your body starts producing lots of ATP for them to go and contract. And so therefore that's why calcium is very important. And you can see that in the picture here. If you have the phosphate here, you get the inactive form. If you don't have the phosphate, you have the active form. So the kinase makes it inactive. This also links it to the energy state of the cell. If you have lots of ATP, it's more likely to be inactive. That makes sense because you have lots of ATP. Do you want to make more ATP? Probably not. And so therefore, that makes a lot of sense. Schematically, you can see the effectors here, ATP, acetyl-CoA, and NADH all negatively affect PDH. That makes sense. If you have lots of acetyl-CoA, why make more? If you have lots of NADH, again, why make more? The same thing for ATP. On the other hand, ADP and pyruvate are going to enhance its activity. Shown here is a little more schematic for the whole pathway. In this case here, we look at pyruvate dehydrogenase. Pyruvate dehydrogenase has similar things to the alpha-ketoglutarate dehydrogenase in the sense that you have ATP, succinyl-CoA, and NADH. Succinyl-CoA is the product for the alpha-ketoglutarate. Acetyl-CoA is the pyruvate dehydrogenase product, so that makes sense. The isocitrate dehydrogenase is regulated again negatively by ATP and NADH. Again, if you have lots of them, why make more? And it's stimulated by ATP for the same reason as pyruvate dehydrogenase was stimulated. And these are the key control points for the citric acid cycle. The other thing about the citric acid cycle that's important to realize is that it also is very important for making biosynthetic intermediates. As shown here are some of the biosynthetic intermediates you can make. I just realized that my time is up for today. So next class, we will finish off chapter 17. We'll do the second worksheet for chapter 17, and we will start chapter 18. So please, for next Tuesday, I can't remember when I set the learning curve. I will set it for anyways for Sunday or Monday. And please do the learning curve for chapter 18 so that you're prepared for that particular section. So thank you all very much today for listening. I was hoping to go through the exam once more, but oh, the gluconeogenesis worksheet. Shoot, I forgot that. Yes, remind me at the beginning of next class, and I will certainly go through that too. Sometimes I'm forgetful, just like everybody else. So remind me, and I will certainly do that next class.
Professor, are the practice exams already posted?
Yes, for the practice third exams, all the former third exams are posted already, or at least they should be. If they're not on Canvas, let me know. Are they on Canvas? Okay, good, they are. Yeah, they were posted on Tuesday. Oh yes, I did get quite a few emails. I've answered some of them, but I haven't quite answered all of them yet. I will get to them today. But the emails, mainly the ones I didn't answer, were the ones concerning with the exam. Yes, how we'll be able to draw mechanisms. Part of the thing to do, first of all, is sort out what technology people have. And then from there, we will go and look into mechanisms on computers. We may not do that. We may be able to do it some other way. Is a past homework going to be available for practice? We should be able to look into that, yes. I know one of my colleagues who teaches physical chemistry is using a program called the CAM scanner, and he's found that to work very well for going and doing written material. Oh, my cat's unhappy. Okay, thank you very much, guys. Have a great day.